Hey, everybody. So we are going to look at chapter 3. Let's give it a second to load here. Chapter 3 is going to be chemical reactions and energy. So it is going to be a little bit of a review from our Bio 101 courses, but it may be a little bit of time since you've taken those, or, the concepts of cell respiration and chemical reactions were kind of hard because it was new then. So this will go through it in some depth. This might be a chapter that you need to take a little bit of time to go through the resources and to, watch this video multiple times, or it may not be. Some parts of it are going to be more in depth than 101. Some parts are going to sort of breeze through parts of 101. So it'll really just be up to you what you need from it. But let's go ahead and dive on in and get started. So, the first thing you'll see is that there are only two objectives from our class syllabus here, and that is explain chemical reactions in the human body and explain metabolism and turnover of organic molecules. Now, that would make this seem like it would be very easy because those are, two when we've been having, six or seven objectives. But those objectives are really much more detailed than they seem. There's lots of steps and parts and pieces involved in answering them. So, again, this might be something that takes a little bit of time to get all of the nuances. So, just wanting you to know that this is a longer slideshow than some of our other ones because of how in depth answering these questions really are. Okay, now that I've thoroughly scared you, let's just go ahead and get on into it. So, the first thing that we want to remember is that all living organisms require energy. And we'll see energy is just this capacity or ability to do work. And these living organisms are going to need energy to do their everyday things like power muscles, pump blood, absorb nutrients, exchange respiratory gas like O2 and CO2 in the lungs, to make new molecules, and to get ion concentrations across the cell membrane gradient. So, those are just some features in the human body that would require energy. And the way that we do this is that we break down glucose through different metabolic pathways in order to make ATP. And ATP is what is the energy currency of our cells. It's what they use to power these different processes. Again, energy is just the capacity or the ability to do work. And there are two major classes that we're going to talk about. The first being potential energy. That is the stored energy, and sometimes it's called energy of position. And if you need an example, easy to look at in your, classroom, home right now, if you have a pen, just hold it above your head. That pen now has potential energy. If you let go of the pen and it starts to fall, it now has kinetic energy. That's the energy of motion. And we'll see from our laws of thermodynamics and physics that we don't make or destroy energy. We just convert it from one kind to another. And that way we're conserving the energy. We're just changing the way that we see it. Like our pen went from having potential energy to then falling. That's kinetic energy. So, if we're looking at the human body specifically, where are we finding these different types of energies? Well, potential energy we find a lot at the plasma membrane. So, there will be a concentration gradient of different solutes, usually an ion, where it'll be more available on one side of the membrane than the other. And that concentration gradient is holding that potential energy. And of course, we know that our plasma membrane, that's just the cell membrane. It's the boundary between inside and outside of the cell, in case you forgot what that meant. And then potential energy, we see that in electron shells. So we've got the information from our last chapter about the octet rule and the number of electrons in different shells. Well, we're holding on to potential energy there as we're moving those electrons from high to lower energy shells, as we're sharing or giving them away in chemical bonds. And then, of course, we want to then change this to kinetic energy, and that kinetic energy can do work. So, again, it's very important to know that we have to change that potential energy that is stored in those concentration gradients or stored in those chemical bonds, and we need to change it into that energy of motion, kinetic energy, so that it can do work in our cells. And here is a diagram that comes from your textbook, just sort of showing you in picture form what we just talked about. So on the far left, we've got this concentration gradient. You can see those phospholipid bilayer, the little things that look like little circles with the little tails. That's your cell membrane. And we can see a channel protein, that's the blue thing. And we can see that there are a lot of sodium ions outside of the cell, and they're falling through that channel protein in order to create or establish this concentration gradient. So we go from potential energy outside of the membrane. That movement of the ions is kinetic energy, and a cell can harness that, hold on to it, and then do other work. On the right picture, we've got an electron transport chain. As we're moving electrons from a high energy to a low energy, we are giving off um, energy that we can harness as work. So we're storing the energy in those electrons as they're in a higher shell. And as they move down, like they fall down the steps, then we're releasing that kinetic energy that can do work. There's some other forms of energy that we need to talk about as well, specifically chemical energy. It's a type of potential energy where we're storing energy in chemical bonds. And then we break those bonds to release that energy. And then we can use that released energy to do movement, to make molecules, or to make those concentration gradients. Chemical energy is present in all chemical bonds. We, of course, talk a lot about it with our ATP, but we can find it in triglycerides, so fats, and glucose, sugar. We're storing the energy in the bonds. And then as we grow those triglyceride fatty acid chains, or we grow glucose chains, if we break them apart, we release energy. Some types of kinetic energy that we see in the human body. We have electrical energy, which is moving charged particles. And a great example that we see is either ions moving across that plasma membrane. When we are talking about neurons specifically, that electrical pulse is an electrical energy. We also see mechanical energy, which is going to be exhibited by objects that are in motion because of some sort of force. And we see this when your muscles contract, which allows you to walk or to move your body. There's sound energy, which is compressing molecules because of vibration. And we see this in sound waves that cause a vibration that hit your eardrum in your ears, which then is what allows you to hear things. There's radiant energy, which is the energy of electromagnetic waves that is being like given off. And we see this as visible light striking your retina, and that is what allows you to see. And then there's heat energy. This is also a type of electromagnetic wave, but heat energy doesn't really have the ability to do work. It's often a byproduct of work that has been done, and it is measured in temperature. So here's an example from your book as well, sort of showing you the different frequencies on electromagnetic spectrum. Do not feel that you need to memorize the different wavelengths. That's not really what this is for. It's just showing you that we have an electromagnetic spectrum that goes all the way from gamma rays that have a very high electromagnetic energy. They can actually mutate our DNA, to x rays that we use in diagnostics, to the UV light, which can also cause damage to cells. Then the visible spectrum of light, where we can see colors, and that light will go to your retina, causing, a chemical reaction so that you see. And then, of course, infrared and radio waves at the lower frequency. But all of these are types of energy, right? It's moving as this wavelength. You can't talk about energy without talking about thermodynamics. We're very briefly going to go through this. This is more of a chemistry, physics type subject. But again, you can't have energy and chemical reaction talk without this. So, thermodynamics is the study of energy transformations, how we go from that potential energy that is stored to a type of kinetic energy that can do work. The first law of thermodynamics tells us that we cannot make or destroy energy. We can just change the form that it comes in. So we're not making a whole brand new set of energy when we take those chemical bonds and break them. We're just turning that stored energy into a type of kinetic energy, into some sort of ability to do work. The second law of thermodynamics says that when we transform that energy, we're going to lose some of it. This isn't a perfect machine that we can conserve all of that energy that we've created. So the usable energy is decreased at each step. So, pretty much an example for your human body, you're cold and you start moving around or your muscles start shivering. That is taking, the chemical energy that you have stored in glucose and the food that you've eaten, and then you're breaking it apart to do that motion, and your body will create heat. It loses some of that energy that was stored in those chemical bonds as this process of breaking it down happens. When we talk about energy and this thermodynamic stuff, we have to talk about metabolism. Metabolism is just all of the reactions that are happening in a living organism. We look at it usually as chemical reactions because that's ultimately what's happening. And it is what's going to happen when we break apart chemical bonds in existing molecular structures to make other new bonds. And we write this as a chemical equation. And chemical equations have reactants. Those are the things that are going to either be put together or broken apart. We see them normally on the left side of a reaction. And then we have products. That's the thing that we make, whether it's a molecule that is bigger because we've combined things, or it's smaller molecules because we've broken them apart. And that's usually what we see on the right side of a chemical equation. So again, let's take a little look here and see our reactants. That's the stuff that we start with before a chemical reaction. And again, we write that on the left side. Then we've got our products. That's what we make. That's on the right side. So in the little example here, we've got A plus B yields C. That little arrow, you read it as yields. So A plus B, that is going to be the reactants, thing A and thing B. That's what you start with. And then C is our product. That is what we make because we've combined A and B. And the arrow is going to show us which direction this reaction happens. When we talk about equations, we want them to be balanced, which means that the number of elements on one side is equal to what we have on the other side. Even if we change where they are located, we want the same number of things. Chemical reactions get classified based on three specific criteria. One, we change the chemical structure, whether that is we break it apart, build, or we switch where things are. We have to change the chemical makeup in order to have a chemical reaction. We change the chemical energy. So we've stored energy in bonds. If we break or form new ones, we're changing the energy. And we classify if it is a reversible or irreversible reaction. Can it go in both directions, or is this a one-way street? And let's start with first classifying chemical structure. We have what we call decomposition reactions, synthesis reactions, and exchange reactions. Those are the main broad categories. Let's first look at a decomposition reaction. This should be pretty familiar with you already. And if it's not the word, you've probably seen this sort of interaction before. It's where we take a big molecule and we break it down into smaller pieces. So what it would look like is you have a substance that we call AB. It yields A plus B. An example is when we do hydrolysis of sucrose and we turn it into glucose plus fructose. We call all of the decomposition reactions that happen in the body as catabolic reactions or catabolism. And I, again, like to think of catabolism. I have a cat. He is very mischievous and knocks things over and breaks them apart pretty constantly. So I think of breaking apart bonds as catabolism. Synthesis is really the opposite of our previous reaction. You take two or more structures, put them together, and make a bigger structure. So now we've got A plus B, and it's going to yield AB. And we can think of this as dehydration synthesis in order to form our dimers or our dipeptide when we're making a biomolecule. And the collective term for any synthesis reaction that happens in the body is anabolism. Then, slightly more complicated, we have exchange reactions. And that is where you've got a group or a chemical that is going to be exchanged between two different chemical structures. So there is a part of this reaction that is a decomposition and a part of it that is a synthesis. And this is what is most often happening in a human body. We're going to take substance AB, we're going to add C, and it's going to yield A plus BC. And the example that your textbook uses, very common, has to happen in our cells, is making ATP. We also want to look at something called an oxidation reduction reaction. It's usually abbreviated as a redox reaction. This is a type of exchange reaction where we're taking electrons from one chemical structure and giving them to another. The structure that loses its electron is oxidized, and the structure that gains is called reduction or a reduced part of the reaction. And we have to have these two things happening together. So, a great example is if we're moving ions with this hydrogen ion. And in your cells, you have NAD plus that then will get a hydrogen and become NADH. And this is happening really in cell respiration, which is why we're discussing this now. We're moving towards that. So here is that sort of reaction blown up for you from a picture from your text, you've got a molecule like glucose, and glucose is going to be oxidized. And again, oxidized is going to be gaining, reducing is the losing. So or I'm sorry, our oxidized, again, over here, oxidize is going to lose, reduction is gain. Sorry about that misspeak. So our glucose is going to give up 2 hydrogen atoms. The NAD plus is reduced, so it gains those hydrogen ions because hydrogen is going to be carrying that electron. It's going to gain it. We also want to classify our chemical reactions as either exergonic or endergonic. Exergonic reactions happen when reactants have more energy in their chemical bonds than the products. So the energy that we have, there's a net decrease in the potential energy. So we go from high energy to yielding something with a lower energy. And that is a decomposition reaction. Endergonic reactions are reactions with less energy in their chemical bonds than the products. So we are going to increase our potential energy. And this example is a synthesis reaction. So what does that look like sort of on a graph? And you will not need to really draw these out and make them, but understanding them is helpful in understanding exergonic and endergonic reactions. So let's look at our exergonic reaction. The first thing that we have here is on our um, y-axis, we have the energy. Do we supply the energy to make it happen, or do we release energy as it happens? We're going to start at 0 because our reactants are our starting place. And then as the reaction happens, that's on the y-axis, or I'm sorry, the x-axis, we are going to be releasing energy because our reactants have more than what we end with. The opposite is that endergonic reaction. Again, we'll start with energy of 0. Our reactants are the starting place. But then as we get to our products, we actually have to put energy into the reaction to make it happen. So we will see that we have a higher potential energy at the end of our reaction. So, what does this mean for ATP and how do we cycle it? What does that look like? So, in your body, we are constantly using ATP. So, we have to constantly make and break it down. I think your book says it's something like 2 minutes or something of ATP is available at any time in your body as a whole. So, you don't have a lot of this ATP for your cells to use built up. We have to constantly make and destroy those bonds in order to keep our cells with energy. We are going to form ATP in these exergonic reactions, and it's going to fuel molecules from food, and those are getting oxidized. Then the energy in their bonds are transferred to ADP and a phosphate that we then can stick them together and form ATP. And then again, we break down the ATP to make ADP. And when we release that, we are getting energy. And of course, there's only a few, oh, even better, only a few seconds at a time in your body. So the formation of ATP has to be continuous in order for your cells to have energy. So here's what that all looks like in a picture form. We've got, we'll start here. We've got ATP. We've got it made. We've got these 3 phosphorus, and we've got this energy bond. It is going to be broken apart and we release energy. That's our exergonic reaction. Then we end up with ADP that we need to have an endergonic reaction. We put energy in and make ATP. And this is a cycle. It just keeps happening by your cells in order to make the ATP and use it for energy. Lastly, we can classify chemical reactions as being reversible or irreversible. So, irreversible reactions, we are going to lose this reactant and gain products. So, we've got A plus B is making AB. We use up A and B, and they're done. At the same time, you could have a reaction that is AB, that then it goes and we make A plus B. But when we use up all the AB, we have A plus B and it's done. We cannot go back and forth. A reversible reaction doesn't just go to the right, like our little arrow pointing that way. It goes back and forth. So there's no net change in the concentration of either the reactants or the products. And then we get what we call an equilibrium. So we can get something like A plus B yields AB, but AB yields A plus B. That's that double arrow. If we increase the reactants or we decrease the products, we can make the reaction happen in one direction. If we decrease our reactants or we increase the product, then we can drive the reaction to happen in the other way. So we can actually change how we are getting our different products or reactants in order to change the rate of reaction. Again, a reaction rate, that is just how quickly a chemical reaction is taking place. Activation energy, that is the energy that we need to break existing chemical bonds. And this is really what is determining how fast or how slow a reaction is taking place. In order to overcome the activation energy, there's a few things that we can do. In a lab setting, you can increase temperature, and that is going to give energy to break bonds. In a human body, it's not always a good idea to increase the temperature because that would denature proteins and that would ultimately stop reactions. So instead, we're going to use what we call an enzyme or a protein catalyst, right? Making it happen faster instead of just heating the thing up. So enzymes are catalysts. Catalysts just accelerate normal physiological activities. An enzyme cannot make a reaction that wouldn't normally happen. It is just making a reaction that would happen slowly happen more quickly. It does this by decreasing that activation energy. We can call a reaction uncatalyzed, there's no enzyme present, or catalyzed, there is an enzyme present. And again, it's very important to note that this only happens with reactions that would already be taking place. We can't make a new, different reaction happen by adding an enzyme. And of course, an enzyme is great because it will increase the rate in which we make our products in a reaction. So, in an exergonic reaction, we've got something like sucrose. Sucrose has a higher potential energy than our products, our glucose and our fructose. It has an amount of energy that is required to start this reaction. That's the activation energy. And we can see here that is this big red line, our uncatalyzed reaction. We have to roll that little ball up that hill in order for our reaction to happen. If we add an enzyme to this reaction, then we lower that activation energy, and it's not as much work to move that little ball up the hill before we get to our products. That is the point of an enzyme. So, what makes an enzyme be able to do its job? What does it look like? Great. Most enzymes are globular proteins. So they are going to be those little fat molecules of proteins that we learned about in our previous chapter. They can range in size from being small, about 60 amino acids, to being really large, complex structures with, over 2,000 amino acids in their sequence. They have a unique three-dimensional shape, and that space is called the active site. That is the place where we're going to make this chemical reaction occur. And what happens is we get our two products, our two reactants in our active site. Then we're able to form this enzyme substrate complex, and then we do our reaction. Then our products are released. So here's what that looks like. We've got this active site on our globular protein. That active site is only going to let a certain type of substrate bind to it. So each chemical reaction that requires an enzyme is going to require a specific enzyme for that reaction. It's not a one size fits all. It's an each individual reaction that can have an enzyme will have its own type. When that substrate binds to the active site, we have this change in conformation or a change in shape, and then we're going to be able to do our chemical reaction there. And when we have this binding, that's our enzyme substrate complex. So, where do we find enzymes? Well, we can find them inside of our cells. An example is DNA polymerase. That DNA polymerase is going to form new DNA and it speeds up that process. We can find them embedded in the plasma membrane. There's an enzyme called lactase that is in the small intestine walls, which are then going to help break apart lactose. And if you are someone who is lactose intolerant, it's because your lactase, you either don't have it or it's not functioning in your small intestines. Some are excreted from a cell. So we've got something called pancreatic amylase, which is going to be released from your pancreas, and that is going to be able to break down starches during digestion. So let's look a little bit deeper at this enzyme structure and how this all works. We call it catalysis. So we've got this substrate that is going to enter into the active site. That makes our enzyme substrate complex. The enzyme is going to change shape and even result in this much closer fit. It's called an induced fit model. There have been several other models, most notably something called lock and key, but we know now that it's this induced fit. It sort of wraps around and gives that substrate a little hug, and we call that the induced fit model. The change in the enzyme shape is going to stress the chemical bonds, which will make new bonds. Then we're going to release our product, and the enzyme can do this reaction again. So here's in picture form what we just talked about. We're looking at lactose, and we've got the enzyme lactase. That's the little blue thing. So we've got our lactose entering the lactase enzyme. We're having this small conformational change that induced fit. We're stressing the bond of our lactose molecule, that bond that's between glucose and galactose. It breaks it apart and we release the glucose and galactose, and now our enzyme is ready to accept a new molecule of lactose. All right, let's take a look now here. This is our glucose, and we want to make glycogen. We want to store the extra glucose. So we can see two glucose molecules fit into this enzyme. Then we're going to stress them out and make a chemical bond between these glucoses, which results in glycogen, this long chain of glucose molecules. That chain is released, and then we are able to use our glycogen synthase again and intake more glucose to add to the chain. Sometimes our enzymes are going to need a little bit of help in order to do their reaction. So we have something called a cofactor. Cofactors are molecules or helper ions that are required to make the reaction happen. These cofactors are non protein and they can be organic or inorganic. If they're an inorganic cofactor and they attach to the enzyme, it's usually our ions like a zinc ion. We call these minerals. So if you take like a multivitamin and you see minerals on there, a lot of them are our cofactors. If the cofactor is organic, we can call it a coenzyme. And these are usually vitamins or some kind of modified nucleotide that act as a coenzyme. Enzymes are lumped into 6 major classes, and this might be something sort of new. We're not going to spend a ton of time on this, but it is more advanced than what you learned in Bio 101. The first being an oxoreductase. That is going to be an enzyme that works in an oxidation reduction reaction. And a great example of it is dehydrogenase, and that is going to be obviously working with hydrogen ions in order to do oxidation reduction reactions. Whew. Then there is transferases. So that is enzymes that are going to transfer atoms or molecules between chemical structures. And we are going to mostly look at kinases that help to move atoms or molecules and really help in motion at a cellular level. We have hydrolases, isomerases, ligases, and lyases. And those all have different functions. Again, feel free, the book gives you a lot more detail, but just knowing that they exist is going to be okay. How do you name an enzyme? Well, it's normally named for either the substrate or the product. They often do have some sort of subclass to them, and we usually use the suffix ase, ace, at the end of it to sort of tell you what it does. So we've got something like pyruvate dehydrogenase. That's going to move hydrogen from pyruvate. So the pyruvate and then dehydrogenase, moving hydrogen from it. Or something like DNA polymerase. So DNA polymerase is used to take our nucleotides and our sugar bases and put them together, so making polymers to make DNA. Lactase breaks apart lactose. So you can see how you got the ace at the end, but we're working on the sugar lactose. What can we do to slow down or speed up or change reaction rates? Well, the amount of substrate or the amount of enzyme can affect how fast the reaction happens. So we can speed the reaction up if we increase the amount of enzyme or we increase the amount of substrate concentration. But we can only increase it until we reach a saturation point. And saturation point means that every enzyme is full of its substrate. And even if we added more substrate, there wouldn't be enough enzyme to continue the reaction until we use it on whatever is binded already to the enzyme. So we reach a point where we just can't make it happen any faster. And that's here. We can increase the reaction rate so much. If we increase the amount of substrate, we increase the reaction until we get to a place where we can no longer speed it up without having more enzyme available. We can also speed up the reaction if we change the temperature. We want to be careful though, because enzymes are proteins. So we've got this three-dimensional shape that is dependent on temperature. Most human enzymes are going to function best at body temperature, like 98.6 degrees Fahrenheit, 37 degrees Celsius. Some will work a little bit better with a slight fever, right? A low grade fever can speed up chemical processes. But if we increase the temperature too much, like we have a high fever for a long period of time, that's going to denature the enzyme and then we lose its function. And we know from our previous chapter, once you denature the protein, it doesn't regain its shape, it's lost. So that can be very serious, resulting in very serious illness and even death in a human when we lose our enzyme function. So again, we can see here, we can graph this on a graph where we've got the rate of reaction on the Y, temperature on the X. We see the shape of our protein, and we can increase it if we increase the temperature a little bit, even a little bit past normal body range. But if we get too far past that, we've denatured the protein, and now we can no longer use it. And that is a cause for concern. pH also has an effect on enzymes. There is an optimal pH or a best working pH, and that's usually between 6 to 8 on the pH scale for most enzymes. And you'll notice like that's where our blood pH normally ranges. That 6 and that 8 are kind of danger zones, but within that range. What happens is that we can change the hydrogen ion concentration, which will mess up those interactions on our enzyme. And of course, if we go too far acidic or too far basic, then we're going to denature our protein and, of course, lose its function. That being said, that a lot of enzymes work between that pH of 6 and 8, and we find that in our blood. There are enzymes that work in very different conditions, like the enzymes that are working in your stomach. They are obviously going to have an optimal pH that is much lower, right? Because our stomach acid is in that 1 to 2 pH range. That's where those enzymes will work. And again, here's what we got going on. If we've got too acidic of an environment, we denature the protein. If we have too basic of an environment, we denature the protein. But at that optimal range, usually between 6 and 8 for most of the body's enzymes, we have enzymes that will work and we don't denature that protein. All right, let's look at some ways that we can control these reactions. So, first, we're going to talk about inhibitors. Inhibitors will bind to the enzyme and stop these chemical reactions. We would want this because we don't want to produce too much of a certain product. That could also be a problem in our body. And then we can later release the inhibitor off of our enzyme and allow this chemical reaction to keep happening. And inhibitors come in sort of two different flavors. They're either competitive or non competitive. So, a competitive inhibitor is going to resemble or mimic the shape of the substrate so that it can bind to the active site. And while it's binded, there's no chemical reaction really taking place, like if it were the substrate, but it's going to compete with the substrate and not let it get into the active site. So you can think of it sort of like two runners in a race. The competitive inhibitor, it's faster. It gets to that active site and it stays there. And now the substrate can't get in there in order to have the reaction happen. If we increase the substrate, then we will have a less likely occurrence of competitive inhibition, right? The more substrate you have, even though if the little competitive inhibitor can get into that active site, if we've got a lot more substrate, it will still beat out that competitive inhibitor. If we have less substrate, well, then that's more likely that our inhibitor will get into the active site and block out our substrate. Non-competitive inhibitors are going to bind to a different site that we call an allosteric site. Once that non-competitive inhibitor is binded, well, then it is going to change the shape of the active site, and now the substrate doesn't fit. These are also called allosteric inhibitors in some literature and videos that you watch. It means the same thing as non competitive inhibitors. And because it changes the shape of the active site, no matter how much substrate you add, it's not going to change the non competitive inhibition. Okay, so now that we've just done all of that, I know it's a lot. What is the purpose? Well, we want to look at metabolism and using multiple enzymes in order to have chemical reactions take place. So, you can call those multiple enzymes in steps a metabolic pathway. And we're going to have a series of enzymes working together in order to break apart or build some other chemical or molecule. An example is breaking down glucose. The product of one chemical reaction is the substrate of the next chemical reaction that is taking place. And we can have these enzymes grouped together and attached in order to have these reactions take place in a sequence. And an example of that is our pyruvate dehydrogenase that we use in cellular respiration. So, what happens here with our multi enzyme complex? It's really great because it's less likely the substance is going to move away or go into a different biochemical pathway. In a single complex, we can be regulating it rather than each individual enzyme, right? Because we need the product to go to the next step. If we can regulate one step, then we stop the whole pathway, as opposed to individual enzymes, each needing their own regulation. Pathways are usually regulated through negative feedback, which means that the product from one of these pathways is going to be the inhibitor in the other steps. So it's going to turn off the enzyme early in a pathway. You can get more product or less product. And as we accumulate that, we can either move on to the next step or we stop the next step. So you kind of think of it like a little gateway or like a series of locks. Like if you are familiar with boating, right? You go into the lock, the water level rises, there's a stopping point until you can open that gate and go to the next series. Same thing with our pathways here. So here's how it works. We've got our first enzyme that needs a substrate. It's going to make something, and that something that it makes goes into the second enzyme, which makes something that goes into the third enzyme, which makes something that becomes our end product. Now, we can keep this pathway open and going as long as we need to make that end product. But if we start to accumulate too much of it, then that end product can stop enzyme number one. It becomes the allosteric inhibitor. And then we don't keep this pathway going until we need more of that end product again. And then we can release that allosteric inhibitor and we can start the process over. So, how do you regulate enzymes? Well, you can have phosphorylation, so adding a phosphate to the group. And we do this with the kinases. And adding a phosphorus can turn some enzymes on, but turn other enzymes off. It just depends on what the pathway is. Then you have dephosphorylation, which is removing that phosphorus that you've added. We do that with something called phosphatases. And again, that will turn some enzymes on to do their metabolic pathway. It will turn others off. If we've got an exergonic metabolic pathway, the organic molecules that are oxidized and broken apart by a series of enzymes, that potential energy that is in those chemical bonds get released, and that energy makes ATP. But of course, we need oxygen present to do that. So, what are these steps here in getting ATP? Well, we need glucose oxidation. We're going to break down glucose, and that breaking down is going to release energy. From the breakdown of glucose, we get carbon dioxide and water. And here's what we know. We know glucose is an energy rich molecule because it has a lot of bonds, specifically the carbon to carbon bonds, carbon to hydrogen, and carbon to oxygen. Our chemical equation that you looked at in Bio 101 is going to be our glucose molecule plus 6 oxygen is going to yield 6 CO2 and 6 water molecules. How does this happen? Well, we're going to break down bonds that are used to attach phosphate groups to ADP. That energy can be used directly to do something, sort of the least common method in the human body, but we call that substrate level phosphorylation. The energy can be used indirectly, which is much more common. The energy is first released to a set of coenzymes. Then that energy is moved from the coenzymes to make ATP. And that process we call oxidative phosphorylation. Where does it happen? Well, there are 20 or so different enzymes required. Those enzymes are found in the cytosol, which is this sort of semi-fluid, goopy, jelly-like stuff in a cell, and in the mitochondria. That's the small organ that is what we call the energy or powerhouse of the cell. Sort of shaped like a little jelly bean in most diagrams that you'll see. So here we go. We got glycolysis. Glycolysis is happening in the cytosol. Then we've got this intermediate stage or the prep reaction, the citric acid cycle, and the electron transport chain, all happening within the mitochondria of the cell. So, what are our steps? Well, first, we've got glycolysis happening in the cytosol. It does not require oxygen to happen. It will happen even if we do not have oxygen. Then you've got the prep or intermediate stage. You've got the citric acid cycle, and you've got the electron transport chain. These steps happen in the mitochondria, and there must be oxygen present in order for them to happen. So let's look at each step a little bit more in depth. Glycolysis, again, can happen without oxygen. It's going to need 10 enzymes in the cytosol to make it work. You will not need to memorize all 10 of the enzymes. Just get that out of your mind now. But knowing that there are 10 and then some of the ones we specifically look at will be very helpful. The glucose is broken down into 2 pyruvate molecules. So our net production here is 2 ATP and 2 NADH molecules. So here's what's happening. Glucose is split into 2 molecules of G3P. We've got to put some ATP in order to end up with our products at the end. So we're going to have these phosphate groups that are going to transfer to break down the glucose. So in this, you're not really going to need to know any of these individual enzymes at this step. Just know glycolysis, you must put ATP in order to break down the glucose and make your G3P molecules. Okay, the next steps is going to have two glucose oxidation reactions. You're going to take this unattached phosphorus and two hydrogen atoms, and those are going to be released to the NAD. The phosphorus is going to be moved to ADP and make ATP. Again, at this stage, you don't need to know any of the names of these enzymes that are doing this, but it is maybe helpful to remember kinase, right? Kinase is moving those hydrogens, so it's maybe useful for you to remember that a kinase is involved here. Okay, the next steps, basically, we're having these two glucose oxidation steps. We're going to take a molecule that we've made and make an isomer from it, which is just a molecule that has a different shape but the same number of molecules or atoms in it. We lose our water from this step, and then of course we make ATP. Again, here we've got this process occurring in the cytosol. It does not require oxygen. You start with glucose, you end with pyruvate. You're going to be using 10 different enzymes, and it's going to end up with putting 2 ATP in to start, making 4 through these processes. So you make a total of 2 ATP and 2 NADH. How do we regulate glycolysis? Well, of course, it's through negative feedback. ATP can act as an allosteric inhibitor to turn off the enzyme phosphofructokinase. It's okay to just know ATP is an allosteric inhibitor to turn off an enzyme. And as we get more ATP, we're stopping this enzyme. The fate of what happens to the pyruvate you make depends on oxygen availability. If there is oxygen, it will go to the mitochondria to continue on into the prep reaction or intermediate reaction, a citric acid cycle, and electron transport chain. If there's not enough oxygen, it's going to be converted into lactate, which happens by fermentation. So let's look at this intermediate stage or the prep reaction. This is what happens right after we go through glycolysis, and it's going to be the remaining stage in the mitochondria. So in the mitochondria, you've got two membranes on this organelle. The inner part that's all folded and bended together is called cristae. There's a space between the membranes in that outer compartment, and the inner part of it is called the matrix. In the matrix, that's where we've got these multiple enzymes that are working together in order to do their reactions. And we also have the enzymes that will do the citric acid cycle here. Molecules in the electron transport chain are embedded into the membrane of the cristae. This intermediate stage is going to link glycolysis and the citric acid cycle. This is where you should know that we have this enzyme, pyruvate dehydrogenase. It is going to take pyruvate and something called coenzyme A, do a reaction to make acetyl-CoA. During this decarboxylation, a carboxyl group is going to be released. And that is going to be where we get our CO2. Energy is released from NADH, and we form NAD+. The acetyl-CoA is what is going to go into the citric acid cycle. And we need 2 turns of this intermediate stage in order for this to then proceed on. So we get 2 NADH from each glucose molecule that goes in. So again, glycolysis happens in the cell. We end up with pyruvate. In the intermediate stage, we use our pyruvate dehydrogenase in order to get acetyl-CoA. That acetyl-CoA and the release of CO2 is what is going to end up going towards the citric acid cycle. The citric acid cycle is going to have 9 enzymes. You don't need to know which ones they are exactly. You're going to take acetyl-CoA and you are going to break it apart to release 2 CO2 molecules and a coenzyme A. And from it, you're going to make ATP, 3 NADH, 1 FADH2 in one cycle of this citric acid cycle. So what are the steps? Coenzyme A gets combined with oxaloacetate, and we form what we call citrate. In the next 2 steps, we're going to remove water and reattach it elsewhere and make isomers of that citrate. Steps 4 and 5 will actually move a hydrogen to NAD and make NADH. And we've got our coenzyme A attached. We remove coenzyme A, that's going to go back to the intermediate stage, and we're going to make ATP. Our dehydrogenase is going to move hydrogens and make FADH2. We release water, and dehydrogenase is going to transfer hydrogen and form NAD. These hydrogens are where we're going to get our electrons that we're moving in our chain. So really we're building up this stored energy as we're making these FADH2 and NADH. So this is just the picture from your book. You do not need to memorize all these shapes and enzymes that are in this and each part, but just knowing what we get out of this is very important. But if the how we get it out is helpful to you, then please dive into this diagram. So summary, it happens in the mitochondria. We need oxygen. We start with acetyl-CoA. We are going to get 2 CO2 and 1 coenzyme A produced. That CO2 is released. Coenzyme A can go back to our intermediate or prep reaction. We've got oxaloacetic acid involved in the first step and regenerated in the last step. That's why it's a cycle, so we can keep going. And we need 2 turns for 1 glucose molecule to be broken down, and we'll get 2 ATP, 6 NADH, and 2 FADH2. How do we regulate this? Well, the first step, we have something called that citrate synthase. And that is where we can stop or regulate our citric acid cycle. If we've got a high energy demand, like we need to be making energy, then the levels of NADH and ATP in that pathway are going to be low. And so we can increase this activity. If the energy demands are low, we're going to have more of our substrate and the cycle will slow down and decrease. And these adjustments maintain homeostasis, and they're happening, even right now. So that is keeping that balance. Last but not least, we've got the electron transport chain. It's going to transfer the electrons that are stored in our NAD and FADH2. And as they fall down this chain, they're going to make ATP. So what is going to happen in our transport chain? We've got this inner membrane in our mitochondria. There is a hydrogen ion pump that is moving hydrogen ions from the matrix to the outer membrane compartment, and then they are falling back through because of this gradient. There are the electron carriers, that's the NADH and FADH2, that are going to be transporting these electrons between these pumps. And then, of course, there's these series of pumps and electron carriers that all together we call the electron transport chain. How it all works. So we're going to have electrons that are transferred from the coenzymes to oxygen. The coenzymes release hydrogen and are oxidized. The electrons pass through the electron carriers, and then the O2 combines with the 4 electrons and the 4 hydrogens and make 2 molecules of water. There's this protein gradient that gets established as we move electrons through the chain, and we can get kinetic energy by harnessing these pumps. The proton gradient is what ends up making the ATP. Hydrogen moves down the gradient from the outer compartment to the matrix. As it is falling, that energy is going to power ATP synthase, and that ATP synthase is what makes the bond between ADP and the phosphorus to make ATP. We call this oxidative phosphorylation. Oxygen is the final electron acceptor. ATP is what is going to be made when we phosphorylate the ADP. We give it a phosphorus bond. This is different than substrate level. The substrate level is going to be ATP from energy released from the substrate, like from the bonds there. This oxidative phosphorylation is this electron transport chain. Substrate level is happening in glycolysis and citric acid cycle. That's where you get the ATP molecules there, and you don't get as many. But in our electron transport chain, with this oxidative phosphorylation, we're making much more ATP. And this is the diagram from your book, just showing you how the proton pumps are working to get the hydrogen gradient. And then as it falls back through, we're able to connect a phosphorus to an ADP to make ATP. And this is a diagram of an entire summary. I like this diagram. It's great. It gives a really good overview of where you're making ATP and how much and what is carrying the protons. I will also say that I've put a really good video of this on the D2L in your extra resources that I think is a better use of this because it'll walk you through the diagram and the steps to show you where everything's happening. So, how much ATP are we making? It depends on where these electrons are in this transport chain. So, electrons from NADH enter sort of at the top of this pump. They get passed through 3 different pumps and make 3 ATP molecules. FADH2 enters at the second stage and only makes 2 molecules. As a whole, here's what we've got going on. In a perfect world, ATP from the breakdown of glucose looks like this. Glycolysis is going to have 2 substrate level phosphorylation and 2 oxidative phosphorylations, and we're going to have 6. Intermediate stage, we just have this phosphorylation happening oxidatively. Citric acid cycle, we're getting 2 at substrate level. When we take our 6 NADH that we have, we get 18 ATP. When we take our 2 FADH2, we get 4. So total here, we are making two total substrate level phosphorylation ATPs, 34 at the oxidative, the electron transport chain level. Total, our ATP needs to be used in respiration. So the net number that we make is 30 ATP when a human body breaks down glucose. That being said, that magic 30 number is sort of a ideal number. We can definitely make less in a real situation. But 30 is this net max ideal number of ATP that are formed. If there's not enough oxygen, then the pyruvate will go to fermentation. And we have much less activity in our electron transport chain. So we're going to actually accumulate NAD and FAD2. We're going to have less NAD plus and FAD. So we have less ways to move those hydrogens and move those electrons in the electron transport chain. As a cell becomes dependent on glycolysis, like we're waiting in this stage of not having enough of our electron transport chain molecules, then we're going to require more of this NAD+. Glycolysis eventually shuts down because we don't have enough NAD, and we've got to make more to keep glycolysis happening. How do we do that? Well, fermentation. So we're going to transfer the NADH to pyruvate. Pyruvate makes lactic acid. And when we do that, we're going to get back our NAD plus that we can add hydrogen to. And we only make 2 ATP, which is not much, but it allows our cells to keep some energy source. And of course, there is an impact with this decreased ability to get oxygen to cells. And we see this fermentation happening if there are individuals with respiratory or cardiovascular disease. And so we'll see a lower energy level because we're not able to get ATP at a cellular level because there's not enough oxygen. We can also bring other molecules in to do cellular respiration. We break down fatty acids, and that makes the acetyl-CoA that is going to go into our citric acid cycle. Amino acids have many different pathways when we are breaking down proteins, and that point of entry is going to depend on which amino acid we're breaking down. When we do this, we always get this amine group, it's a nitrogen group, as a waste product that is made into urea and excreted by the kidneys. And I know that is a lot and a lot of very technical information, but really, if we just get those main concepts of 30 net ATP, that most of them are coming from this oxidative phosphorylation, which is our electron transport chain. If we're getting that fatty acids and amino acids can also enter this process, if we're getting our basic types of energy, then we're good. But there is a lot of smaller steps in here to describe the reactions happening. So of course, take some time going through the book and through the Connect resources and through the PowerPoints, through the review guides, and hopefully it makes sense. There's videos as well. And of course, feel free to reach out to me if you have specific questions. Otherwise, thank you for watching this video, and I hope that you guys have a wonderful rest of your day. I'll talk to you next time.